Mr. President, Maurizio Brunori, thank you very much for your kind introduction. It's a great honor for me to come today to give this lecture in memory of Max Perutz. You've heard that he was the founder of the Laboratory of Molecular Biology in Cambridge, where now, since the foundation in 1962, 14 Nobel Prizes have been awarded. The latest one this last year to Gregory Winter. I was number 11. And Max himself was a Nobel laureate in 1962 for his work, as you've heard, on solving the structure of hemoglobin. And here he is, late in life, with his beloved hemoglobin molecule. So I'm going to talk about a different topic, as you know, the fuel of life. And the first question that I want to ask is, where does biological energy come from? Because all life requires a constant supply of energy. And largely the answer is that biological energy comes from the sun, where, of course, to a first approximation, it's produced by nuclear fusion of hydrogen to helium. Well, that's a simplification. There's another source of biological energy, and that's geothermal energy. So here's one of these thermal vents deep in the oceans, and there the energy is coming from radioactive decay in the mantle of the Earth, and also probably there's residual heat from early events of planetary collisions leading to accretion. And although this seems a rather inimical environment deep in the oceans, devoid of oxygen, in fact, it's able to support abundant life. As you can see here on the right, there are tube worms and crabs and a very diverse flora and fauna in this seemingly unfriendly place. And of course, they are dependent upon a supply of methane to live. Now, we're, of course, in everyday life familiar with the idea of harvesting solar energy through these solar panels. Another way of harvesting solar energy is through hydroelectric dams. So the sun heats the oceans evaporate, the evaporation forms clouds, the clouds go over mountains, they precipitate rain, and then we collect them in these dams. And we use the potential energy then to convert to drive turbines, as depicted here, to generate electricity. Now, I mention this particular way of converting energy because there is some relationship, as you'll see in a moment, to what happens in biology. Now, the way that energy is harvested from the sun in biology, of course, is through the process of photosynthesis in green plants and algae. And this is a very complex process biochemically speaking, but now the reactions are understood very well in great molecular detail. Possibly the most important reaction on Earth is the splitting of water, two water molecules, to produce oxygen and reducing equivalents, as depicted here. And the other part of photosynthesis are the so-called dark reactions, where the reducing equivalents are recombined with carbon dioxide to produce carbohydrates. So this very complex biochemistry is going on in the chloroplasts of green plants, and we now have a very good understanding in chemical terms of what is happening. And of course, we harvest solar energy on an industrial scale by growing crops. Here are wheat fields in the Midwest of the United States. And we then consume the crops, the carbohydrates in the crops, either directly by eating them or by feeding them to animals indirectly, then consuming the meat from the animals. Now, what about what happens in the human body? So here's a depiction of ourselves here on the left with the organs shown. And the organs are made from cells. They're bounded by a membrane, phospholipid membrane, and the cells, with the exception of red blood cells, contain a nucleus where the DNA is. And then in the cytoplasm of the cell, there are organelles. So there's a Golgi apparatus, the endoplasmic reticulum involved in synthesis of proteins, lysosomes, which are kind of garbage disposal for mopping up proteins that are misfolded or damaged in some way, and then the mitochondria. So I'll now focus on the mitochondria because the mitochondria are the cellular powerhouses, and it's here that the most important reactions in biological energy conversion occur. Now, about the time when I said 25 years, actually 20 years it took to solve this structure. When I started to work in this field in 1978, so I was inspired actually by the work of Frederick Sanger, whom Maurizio has described. So one of the things that Sanger did early on, once he'd learned how to sequence DNA, was to sequence a small DNA molecule that each of us has that is actually found inside the mitochondria. And it's a very interesting molecule, very small in relation to the massive amounts of DNA in the nucleus, only 16.5 kilobases of DNA. But it has the interesting property that it's inherited almost uniquely from our mothers. So there's very little, if any, paternal component in mitochondrial DNA. And it was through Sanger's work that I became interested in what are the mitochondria doing and what are the energy converting processes, and could I contribute in some way to understanding those processes. So at the time, this is the way we thought of mitochondria. There's an electron micrograph here in negative stain. You can see it's bounded by a double membrane. There's an outer membrane and then a highly invaginated inner membrane. And it's here where I'm going to focus, because here, this is where, in these inner membranes, this is where the energy converting processes go on. And here's a kind of cartoon of a 3D cartoon. So we thought of the mitochondria as being somewhat related to bacteria, reflecting their probable origin many billions of years ago from a bacterial origin. Now that rather static picture of the mitochondria has changed during this period now of more than 30 years. And here is part of a skin cell. The membrane is here, the nucleus would be up here. And these are the mitochondria in the cell. You can see that they're joined up, they form a reticulum. That reticulum is mobile, it's remodeling. If you watch here, you'll see little bits will bud off, and then they'll refuse back into this reticulum. And in other cellular states, for example, during cell death, during the process of programmed cell death called apoptosis, the mitochondria break up into small vesicles. And so constantly it's moving between the vesicular state and then this fused state depicted here. Now the significant, I just mentioned this in passing, because the significance of these events is not entirely understood today. We understand quite a lot about the processes of fusion and fission, but not the reasons of mitochondria, but not the reasons why it is so. So as you've heard, ATP is the currency of molecular biology, depicted here in a simplistic kind of way. And the mitochondria generate huge amounts of ATP. So each of us needs to generate every day of our lives 60 kilograms of ATP in order to carry out our normal bodily functions, to make our muscles work, and particularly to think. The brain actually consumes large amounts of ATP to generate membrane potentials that are required in the functions of the brain. And the ATP molecule gives up its energy in whatever the process is. Of course, DNA replication, protein synthesis are all examples of essential processes in the cell that require large amounts of ATP. It gives up its energy And the products are adenosine diphosphate and phosphate. And then what we do is we use the energy that we get from ingesting foodstuffs to put the waste products back together to regenerate more ATP. So there's this cycle of synthesis and degradation and resynthesis going on the whole time. And what I'm going to show you is that in mitochondria, these ATP molecules are made by molecular machines. So first of all, there are proton pumps that are involved in harvesting the energy from food. And then that energy is stored as an intermediate potential energy, and it's somewhat analogous to the storage of water in a hydroelectric dam. And then I'll show you a molecular turbine, and this is the ATP synthesizing machine that I've studied. Uses this potential energy to make ATP. Now, the ATP is being made inside the mitochondria, and if it's going to be useful to the rest of the cell and the organ and the organism, it needs to be transported out. And so there's a transport system to deliver ATP to biological sites. And then, as I've just said, hydrolysis of ATP releases energy to do work. Now, if the sphere represents the inner membrane of the mitochondrion, the actual process, the principle of the process that's going on in generating the potential energy intermediate is in essence a very simple one, because all that's happening is that the chemical energy that's come from the oxidation of sugars and foods in fats, sugars and fats in our foods, is used. This chemical energy is used simply to displace protons, hydrogen ions, from the inside space to the outside space. So this builds up an excess of hydrogen ions on the outside. And in so doing, what has happened is that the chemical potential of these protons has been increased. So there's a chemical potential difference for protons across that membrane. And it's that chemical potential for protons that is the intermediate in energy conversion, which is then used in the synthesis of ATP. Now, this was a very strongly debated idea amongst biochemists in the 1960s. What is the intermediate in energy conversion? And 99% of the world believed that there was a chemical intermediate, a covalent bond intermediate. But this concept was developed by a British biochemist called Peter Mitchell, and he called it chemiosmosis. And for this development, Mitchell received the Nobel Prize in 1978. And he developed a formalism he called the chemical potential difference across the membrane. He called it delta mu H plus. And he coined the name proton motive force. And he did that. He coined it by direct analogy with the more familiar electron motive force that we are familiar with in electricity. So in the biological world, there are three different ways of generating proton motive force across the biological membrane. One is the one that I'm going to describe in mitochondria. We refer to that as respiration. The second procedure I've mentioned en passant already is photosynthesis. So during the events of photosynthesis, the water splitting reaction, the proton motive force is an important product. There's a third way that it's generated, and this is through a simple protein called bacteriorhodopsin. Used to be thought of as an oddity. It's present in planktonic bacteria that we find in the oceans. But on a global scale, this is actually a very important mechanism. So what bacteriorhodopsin does is to absorb light energy and use that light energy to translocate protons across a membrane and generate a proton motive force. 5/6 of the Earth's surface is covered in water, and so the number of these planktonic organisms is extremely high. And in global terms, this is a very important way of generating proton motive force. Now, once proton motive force has been generated, you can use this form of potential energy to do different kinds of work. And one of them that occurs in biology that I won't discuss in detail is to generate heat. And this is the basis of a mechanism known as non-shivering thermogenesis, which is an important mechanism in newly born children to help them generate heat and control their body temperature. We used to think that this mechanism disappeared early in life, but actually in recent years it's been realized that actually even we as adults have this mechanism still. We still retain the capacity to generate heat from proton motive force. Now, this is of particular practical interest when we are faced with this medical problem of obesity, because if one could activate this pathway even more, it would be a way of burning off excess energy instead of generating fat, of just being a little bit hotter and producing heat from proton motive force. And another way that proton motive force is used is to actually transport hydrophilic and charged molecules through hydrophobic membranes. So it's the basis of much of membrane transport. And then the other two examples I've shown here are the rotation of the bacterial flagellum, I'll show you that in a moment, and the generation of ATP. Now, Mitchell developed this formalism to describe proton motive force, and he showed that it has two components. One is a difference in membrane potential across the membrane, and the other component is the difference in pH. And in the mitochondria, this is the difference in membrane potential is by far the dominant component. Actually, there's only a pH difference across the mitochondrial membrane, about 0.6 to 0.8 pH units. And therefore, to a first approximation, proton motive force in mitochondria is a voltage. And across our inner membranes of our mitochondria, we generate a voltage of minus 180 millivolts. In chloroplasts involved in photosynthesis, the converse is true. The membrane potential is low, and the dominant component of proton motive force is the difference in pH, and a pH difference of 4 or 5 pH units can be generated by photosynthetic reactions. Now let's come to the first example here, and that's the rotation of the bacterial flagellum. So this is a Salmonella typhi cell, and you can see that attached to it are these long filaments, small number of them. They're rotating in a clockwise sense, and they bundle together to form this superstructure here. And the rotation of this superstructure drives the organism forward through the liquid medium. Now, what happens is that this motor is capable of reversal, and when that happens, instead of forming this superstructure, the flagellae, now rotating in the opposite sense, stand out from the body of the microorganism. This makes it tumble. And so in the absence of an attractant, a food, a source of food, or something that repels the bacterium, the organism is undergoing a random walk where it's swimming forward a little bit, then it tumbles, then it swims off in another direction, tumbles again. But if you introduce a source of food, sugar for example, the bacterium will move towards the food, not by swimming forward continuously, but through a biased random walk whereby the forward swimming motion now is greater than the tumbling motion, and they'll quickly arrive at the source of food. Now, you can see that the flagellae are rotating, and here on the right are details of the rotor. So in the top picture here, they're identified. So in the inner membrane, which you can see better here, there's a rotary device, and that rotary device is driven by the proton motive force. Each rotation requires the translocation of several hundred protons to generate one circle of rotation. And then that rotation is transmitted through the outer membrane of the organism through a bushing, depicted here. And then there's a universal joint here, which then allows the flagellum to rotate. And the important point in the context of this lecture is that this rotation is driven by the proton motive force that the bacterium has generated across this membrane. Now, what happens in the mitochondria? I'm going to give you some details now of the biochemistry very quickly. So sugars that we've ingested in food are broken down by a process called glycolysis outside the mitochondrion, producing the molecule pyruvate. And pyruvate is then actively transported into the mitochondrion, where it encounters an enzyme called pyruvate dehydrogenase, which converts pyruvate to acetyl-CoA. And importantly, a product of this reaction is NADH. In other words, NAD, the oxidized form of this dinucleotide, is being converted to the reduced form. And then the acetyl-CoA feeds into the tricarboxylic acid cycle, also known as the Krebs cycle. I won't go through it in detail. But from that cycle, you can see that there's more generation of this reduced form of NAD at several points, and also another reduced molecule, FADH2. And what happens with fats? So the acyl groups on fats are, first of all, transferred onto coenzyme A, and thence they're transferred onto a molecule called carnitine, producing acyl carnitine. And acyl carnitine is then transported by a specific molecule that was identified in this collaboration with Ferdinando Palmieri as the acyl carnitine carrier. So the acyl carnitine goes inside, and then the reverse happens. So the acyl group is transferred back onto coenzyme A, producing acyl-CoA. So this is an indirect shuttle mechanism of getting acyl-CoA through this membrane barrier. And then, I won't go through the details, but then the acyl-CoA feeds into another pathway known as the beta-oxidation pathway, where the fatty acid becomes oxidized. And again, importantly, out of those reactions come more reducing equivalents here and here. Now, the reducing equivalents, in other words, high energy electrons, then feed into what is known as the electron transport chain. This is made of three very complicated enzymes. I'll show you in more detail in a moment. And what they do is to take the energy from the reducing equivalents provided by NADH. They take the electrons, high energy electrons, they pass them through this chain, and concomitant with those processes, protons are ejected from the inside to the outside, and this is how the proton motive force is generated. Now this shows you the same thing in more detail. So these are the three important proton pumping machines. They're called complex 1, complex 3, and complex 4. So the electrons feed, so the complex 1, which my colleagues and I defined as being made of 45 polypeptide chains. It's a huge enzyme, megadalton in molecular weight. Its structure has been defined by a colleague, Leonid Sazanov, and another colleague, Judy Hurst. And so what happens is that the electrons actually are fed in at the top of this L-shaped structure. This is about 100 angstroms by 100 angstroms, the two arms. The electrons are quickly transferred through the extrinsic arm by quantum electron tunneling processes. I won't go into the details of it. Via, first of all, flavin mononucleotide and 4 iron-sulfur clusters. And ultimately, they transfer to an oxidized quinone, oxidized ubiquinone, in the membrane, producing the reduced form of ubiquinone. And concomitant with those electron transfer processes, 4 protons are ejected. Now, the ejection of the protons requires energy, and at the moment, one of the active areas of research is trying to understand how energy is transferred into the membrane arm to bring about the proton translocating processes. Another way of producing the reduced quinone is to feed in electrons via succinate to this complex here. There's no proton translocation involved, but the electrons are used to generate more reduced quinone. And by this complex, so these electrons are coming from degradation of amino acids. The reduced quinone then arrives at complex 3, and again, without going through the details of the process, more protons are ejected. Oxidized cytochrome C binds in this region here. It accepts the electrons, becomes reduced, loses its affinity for this complex, and then carries them to the terminal complex, which is called cytochrome C oxidase. And here two things happen. The oxygen that we breathed in becomes reduced back to water, and more protons are translocated. So this is how the proton motive force is generated. And it's this reduction of oxygen to water that allows us to calculate the amount of ATP that we make each day from the amount of oxygen we're respiring through our lungs. And then here is the machine that makes ATP that I'll describe in a moment. And then two other proteins carry this one carries the ATP out and brings back spent fuel ADP, and this one brings back the spent phosphate to be fed back into the machine to generate more ATP. So let's come to the turbines that I decided to study in the mid-1970s. Now, up to that point, not much was known. There'd been some brilliant early work from an American biochemist called Ephraim Racker. And Racker, amongst other things, examined the inner membranes of mitochondria in negative stain electron microscopy and could see structures that he called lollipops or mushrooms attached to these membranes. But he could see that it was like a lollipop. There was a spherical head with a stalk attaching the head to the membrane. And he demonstrated that this actually was the protein synthesizing complex. So we now move on 25 years, and this is actually what the mushrooms and lollipops look like. So this is the catalytic part of the enzyme here. It's bound in the membrane through the blue part and the green part. And protons from the proton motive force are going back into the mitochondria, releasing their energy into this structure in such a way as to generate torque and make the central part rotate. And it's the rotation, the turning of the rotor, that carries energy mechanically into the synthesizing part of the enzyme. And you can see that's one of three catalytic sites here. You can see ADP and phosphate going in, they get entrapped. It then opens up and an ATP molecule flies out. There are three such sites, and so every time the rotor goes round through 360 degrees, you produce three ATP molecules. Now the rest of the structure here is part of a stator, so what it's doing is to hold this part static together with this part in the membrane. I'll come to later. It's holding them static relative to the turning of the rotor. And the rotor can be going around anywhere from 200 to 400 per second. So this is really a massive synthesis of ATP that's occurring in mitochondria. Now, I proposed this idea of a rotary mechanism I think it was in 1994. It wasn't universally accepted. People patronized me and told me I didn't understand enzymes. But Japanese scientists did this beautiful experiment. They took, you can see the catalytic part of a bacterial ATP synthesizing complex and bound it to a substratum chemically through covalent linkage. And then, guided by the structure that we determined, they were able to introduce a cysteine residue here at the tip of this rotor protein, and to that they could bind the molecule biotin. And biotin has a very strong affinity for a protein called streptavidin. Streptavidin actually has 4 biotin binding sites. And then they took an actin filament from mouse skeletal muscle depicted in orange here and made it fluorescent by adding a fluorescent dye. And they also introduced biotin molecules into the actin filament so that they could then bind it to one of the other binding sites in the streptavidin. And they then examined the activity of this chimera in an optical microscope. The point being that the actin filaments are sufficiently long or big that you can actually see them in a light microscope. And then they added adenosine triphosphate, ATP, and observed to see what would happen. And the answer was that they could see actin molecules now rotating like a propeller blade, rotating in this counterclockwise direction as viewed from the chimera that's produced there. Now, clearly, this is not going round at 200 per second. It's going round at about 1 to 2 per second. And the reason for that is actually to do with the scale of the chimera. So, as I'm used to saying, if I represent the enzyme, it's as though I've got a rope 0.5 kilometer long attached to the top of my head. And, of course, the turning of the rope through the aqueous medium, the rotation is slowed down tremendously by viscous drag, and that's why the rotation is so slow. Now, more recent manifestations of this experiment, the actin filament has been replaced by beads, gold beads that are smaller, but you can still observe them optically, and much higher rotation speeds have been generated. The rotation is entirely dependent upon the presence of ATP. If you take the ATP away, the rotation stops. And then they did something else that was very interesting. What they did was to reduce the concentration of ATP, made it less available. And under those conditions, you could see that the rotation tended to stall, and they went on to lower concentrations. And indeed, what they found was it would stall in a particular position, wiggle about, and then move through 120 degrees, stall again, and then move through another 120 degrees, demonstrating that the rotation is not a continuous rotation, but it's a stepping rotation. Now that we can now identify different mechanical elements in this molecular motor. There's a spring, I'll show you in a moment, where energy is stored transiently and released in a quantized fashion. There's a crank, and the crank is modulating as the rotor goes around. It's an asymmetrical rotor. As it goes around, the crank feels the asymmetry of the rotor. And it's these motions in the catalytic sites that bring about the binding of ADP and phosphate, the formation of ATP, and then its release. And then there's a bearing where friction, whatever friction is, allows torque generation. So here's the rotor now. So there's the membrane part turning around, and you can see that this part is stepping through 120 degree steps, implying that in this structure, we think we know where it is, energy is stored and then released in a quantized fashion. And then look at the structure of the rotor. You can see it's asymmetrical. This part here is the part that's making these proteins swing out and change their binding affinities for ADP, phosphate, and then ultimately ATP. Now, one of the current interests is how is torque generated from proton motive force. So now here is in simplified terms, the membrane part of the rotor. It's made of a single protein in multiple copies. They're called C proteins. They're very simple proteins. They have 2 transmembrane alpha helices, and they make 2 rings. The N-terminal helices on the inside, C-terminal helices on the outside. And in the middle of the C-terminal helix is a carboxyl group. and it's right in the middle of the phospholipid bilayer. That tells you immediately that has to be of significance. And then part of the surface of the ring is in association, close association with this green protein, which has, and this is now verified experimentally by structural analysis, which has two half channels. One of them allows protons to go in and neutralize this negative charge on the carboxyl group. Now, once that's happened, the carboxyl group is now more hydrophobic, and so it will move simply by Brownian motion to this position here, bring another negative charge, another proton goes in, neutralizes the negative charge, moves another step around. And so the protons then get carried through the membrane domain until they reach the second half channel, which points into the other side, into the inside of the mitochondrion. And here the protein has properties that makes the carboxyl group reionize and release protons on the other side and generate the negative charge. Now people ask, why is this unidirectional? Surely it requires some kind of physical mechanism to stop it going backwards, but no, that's not required, because there is an enormous pressure of protons from the outside pushing it unidirectionally in this counterclockwise sense, because, as I mentioned, there is a voltage of minus 180 millivolts across this structure here. Now, I said a moment ago, each 360 degrees of rotation, because there are three catalytic sites, and the catalytic part generates 3 ATP molecules. And a full 360 degree rotation requires the binding of 1 proton per C subunit. And therefore this ratio, protons to ATP, is the symmetry of the C ring divided by 3. So if as shown in that picture, I didn't say this, but there were 12 C proteins in the ring, the ratio would be 4. And it was assumed before we investigated this that this would be a universal number. It would be the same throughout biology, but not so. Ring symmetries differ between multicellular and unicellular life, and the ratio is not a universal number. So on the left here at the top is the human ring. It's the smallest. We don't think there will be a smaller ring. It has 8 C proteins in it. This is Saccharomyces cerevisiae, the yeast, has 10. This is a bacterium, 11, another bacterium, 13. This is the ATP synthesizing machine from spinach chloroplast, 14. Here's one with 15. Today there's C9, C10, C11, C12, C14, 15, 16, and even 17. And so what does this mean? Well, I think the way of looking at this is that these are different gears in this machine. This is a high gear, this is a low gear. And in order to drive the high gear, you need a high proton motive force across the membrane. If you can't generate a high proton motive force, then you have to opt for a smaller ring, a larger ring, a lower gear, and generate the rotation in the same way. Now, the energy cost, of course, is the same. It's just the size of the increment that's required each time that you put into the rotary mechanism. Now, that's all I'm going to say about the mechanism of this machine, and I wanted to broaden out a little bit and point out that this topic of bioenergetics, as Maurizio called it at the beginning, is a very important factor in influencing human survival. And we know that defects or that bioenergetics is involved in the aging process, and we have this problem of aging population and growing in size. And then there are also suggestions which as yet are not totally convincing of the involvement of defects in energy generation in mitochondria in neurodegenerative diseases. The most convincing evidence is found in a familial form of Parkinson's disease, where it's absolutely clear that defects in mitochondrial function are associated with that particular form of Parkinson's. With sporadic Parkinson's, and people argue that sporadic Parkinson's and familial Parkinson's are different diseases, it's not so clear yet. And then what I'm going to talk about is the infectious diseases and the increase in drug resistance, and of which upsurge in tuberculosis is a growing major global problem. I could have talked about the increasing demand for energy. I don't have time to do that, and about green chemistry, but I'll save that for another day. Now, in this discussion of bacterial drug resistance, I want to take you back to 1928, to Fleming's famous experiment where he had Staphylococcus aureus growing on an agar plate, that's the streak here, and then a spore of penicillium flew in through the window, landed on his plate, producing penicillin and producing this zone of inhibition, the discovery of penicillin exuded by the mold. That observation was reported in 1928. What happened next? Well, the answer is nothing. Fleming actually did very little to convert his observation into a useful medical drug. And the observation sat fallow in the literature for 11 years until Howard Florey and Ernst Boris Chain, both at Oxford University and the Sir William Dunn School of Pathology, decided to start investigating not just penicillin, but actually a whole range of observations where products were being produced that seemed to have an antibacterial effect, but amongst them was penicillin. And very rapidly they homed in on penicillin. In 1940, 1 year later, Edward Abraham, who was my PhD supervisor, demonstrated that Staphylococcus aureus would become rapidly resistant to penicillin. So this is the first observation of penicillin resistance before penicillin had even been put into clinical practice. That happened in 1943, thanks to the heroic efforts of the scientists at Oxford University. It's a well-known story. And Florey, who was a medical doctor, went to the battlefields in North Africa and treated wounded soldiers with penicillin so that they would recover from their wounds more rapidly and could then return to the battle. And this is considered by some to be an important factor in the events that happened in North Africa. And then in 1945, Fleming, Chain and Florey were jointly awarded the Nobel Prize for Physiology or Medicine for the discovery of penicillin and its curative effect in various infectious diseases. Now, after that, there was a period where many antibiotics were discovered. Sulfonamides had actually come before, and you can go down the list here. This is the year that they were deployed. Penicillin 1943, streptomycin, important in treating tuberculosis, 1943. And you can follow them down the list, all these well-known antibiotics. And the point is that no sooner have they been deployed into clinical medicine than resistance of the organisms is observed within a very short period of time. So what is going on? Well, the answer is simple. So many people misunderstand this process. They think that somehow the antibiotics are mutagenic and they're making the bacteria become resistant. Not so. The resistant bacteria are already present in the population. And the effect of the antibiotics is to kill off the sensitive organisms, the blue ones, and leave the resistant organisms behind so that they can then proliferate in the presence of very few sensitive ones that have survived and ultimately become the population of resistant organisms. So that is what happens. And here are some information about the increase in antibiotic resistance. So the blue curve is multi-resistant Staphylococcus aureus, rising relentlessly from 1981 upwards to the present day. Actually, this only goes to 2001. The green line is vancomycin-resistant enterococci, another pathogenic organism, again rising up, and the red line is fluoroquinoline-resistant pseudomonads. So these are just examples of the kinds of things that are happening in the world outside as people are treated with antibiotics. And this is some more recent information from England. Antibiotic resistance, blood infections, you can see are rising relentlessly, 2013, 14, 15, 16, 17. Now, this one, 2016, is of particular interest to me because among the 15,000 people here, I'm one of them. And it was no fun. So it's now predicted by the World Health Organization that by 2050, deaths from antimicrobial resistance will far exceed the number of deaths from cancer today. So it's going to become, unless we do something about it, it's going to become a huge global problem. And the burden will not be distributed evenly throughout the world. It will be felt mainly in Asia and in sub-Saharan Africa, and be less of a problem in Europe, North America, and South America, and Australia. Now, why is bacterial resistance spreading so rapidly? This selection is going on, but there are factors that we can deal with. So first of all, it's quite clear that medical practitioners overprescribe antibiotics. They give antibiotics under inappropriate circumstances. This then exposes the organisms and selects the resistant ones. The second one I don't understand. And the third one here, which is clearly a major factor, is the overuse of antibiotics in livestock and fish farming. So antibiotics are given to animals prophylactically to keep them healthy. So these are not animals who are ill, diseased. It's to stop them from getting disease. And then there are other obvious things like poor infection control in hospitals, lack of hygiene. And another factor is that in recent years, there's been a lack of new antibiotics being developed. Now, here some data about prescription of antibiotics in daily care. So this is Greece at the top, and then France here, and this is the United Kingdom, much lower levels, Germany, and at the bottom the Netherlands. I'm afraid I don't know where Italy sits in this particular picture, but clearly there are great differences in clinical practice in the way that antibiotics are released to the population. And then this picture here emphasizes the problem in the farming industry. In the United States, currently 70% of antibiotics are consumed by animals and 30% by humans, and clearly this has to change. And then finally, this problem of the discovery gap. So after beta-lactams, penicillins, and the sulfonamides in the 1940s, there was a great interest in finding more antibiotics, tetracyclines, macrolides, aminoglycosides, chloramphenicol, glycopeptides, and quinolines, and so forth. And then after the 1960s, people lost interest in it, and including the pharmaceutical industry, and there was a gap. Nothing happened for 25 or more years. No more antibiotics were found. And then people started to wake up at the beginning of this century, and new peptides, new antibiotics have been discovered. Now, one of the most devastating parts of this problem is tuberculosis. So there's a resurgence in tuberculosis. Sorry. So let me remind you, tuberculosis is a disease of the respiratory system. It's spread to the brain, kidneys, and bones. It's highly contagious via coughing. And the symptoms are chest pain, fatigue, weight loss, and fever. And currently in the world, 2 billion people are infected with Mycobacterium tuberculosis. And in 2017, it was estimated that 1.6 million people died from tuberculosis. And of those who are untreated, so many of the the people who contracted tuberculosis are in the less developed parts of the world, and often they're untreated and they will die. Now, I mean, when I was a child, I can remember the tuberculosis in the village where I lived, and it was relatively common. One of my uncles actually died from tuberculosis. But then that problem went away because of the introduction of antibiotics, and now it's coming back. And why is there this resurgence? Well, one factor is there's actually very poor adherence to TB control programs. So if you contract tuberculosis, the regime for treating it is very long. You have to take drugs for up to 6 months, and that can be very difficult if you're living in a rural part of sub-Saharan Africa. But allied with this is this tremendous problem of the emergence of drug resistance. So there are now multiple drug-resistant strains of mycobacterial tuberculosis, extremely resistant strains. And in the last 3 years, totally drug-resistant strains, that is to say, strains that are totally resistant to every known antibiotic, have been found in India. And there's often a widespread synergy of tuberculosis with HIV/AIDS, because, of course, people with AIDS are immune suppressed. They have very little defense. There's also a problem in quick and accurate diagnosis, and the drugs and vaccines are generally outdated. And there's another problem, and that is that in the human body, tuberculosis is found in 2 different states. It's found in an active state where it's growing and dividing, and there are drugs that will deal with that. But there's a cryptic or dormant state where the microorganisms, they're the green bits here, become surrounded by T cells and B cells, and they form these granulomas surrounded by collagen. So you can take drugs and kill off the active Mycobacterium tuberculosis. You think you've cured the disease, but then the bacteria in the granuloma will emerge through the action of this metalloprotease, releases the bacteria that were surrounding the drug, and the disease is off again. So we need new drugs against novel targets. We need new vaccines. So the vaccine that's currently used throughout the world is called BCG. It was developed at the Pasteur Institute in 1921. BCG stands for Bacillus calmette-guerin. And we need better methods of diagnosis. So here is more information. And 500,000 people have multi-drug resistant tuberculosis. I mentioned 1.6 million died in 2017. And 47% of all drug-resistant patients are in China, India, and Russia. Now here is the most significant discovery in TB research in the last 50 years, published in Science, I think it was in 2008. A diaryl quinoline drug active on the ATP synthase of Mycobacterium tuberculosis. In other words, the ATP synthesizing machine is the drug target for this diaryl quinoline drug. It's a very effective drug. It shortens treatment down to 8 weeks, and it's effective in patients that are drug resistant, and it will also kill the mycobacterium that are hidden in these granulomas. So it's the first anti-TB drug approved in 40 years. It inhibits ATP synthase by binding to the rotor of the enzyme. It's highly specific for mycobacteria. I won't say it has no effect on the human enzyme, but it clearly has a very strongly differential effect. And it's active, as I've said, against dormant and replicating states and against multiple and extremely and totally resistant organisms. However, bedaquiline is not a wonder drug. First of all, the FDA in the United States approved its use based on phase 2 trials with only 440 people. Highly unusual, in fact, I would say unique. Normally you'd have to go on to phase 3 trials, you'd have to involve far more people, but such is the dire need for the drug, they rushed it through. And currently it's recommended only for treatment of multiple extremely and totally resistant strains. And the reason for that is there's a cardiovascular risk. In other words, there are side effects. The drug is very lipophilic, and it persists in the human body for up to half a year, and people become susceptible to phospholipidosis. And they can lead to death. But these people are in dire straits anyway, and 10% of those deaths are unexplained. So what we need are more inhibitors of the mycobacterial ATPase. So what my colleagues and I are doing is to try and contribute to this by providing a basis for rational drug design. So we just determined the structure, atomic resolution structure of the catalytic domain of the enzyme from Mycobacterium sphegmatis. This is not tuberculosis, which of course we can't work with, but a closely related non-pathogenic strain. And we can see differences, and we think there is potential for developing other molecules to intervene in the action of this enzyme. Now, this can be generalized, so all bacterial pathogens have their ATP synthase and they become potential drug targets. But actually, they've been hardly studied at all, except for in E. coli. And bacteria live in diverse conditions of pH, oxygen, salt. They can live anywhere from pH 2 to 10 in the presence or absence of oxygen, in very high salt concentrations, sometimes up to 3 molar salt. Some of them are driven by proton motive force. Others instead use a sodium-ion motive force, analogous to proton motive force. Some of them can both make and hydrolyze ATP. Others, the one in mycobacteria, can only synthesize, and we don't understand that, and that's a very interesting topic of research. The regulatory mechanisms are ill-defined, and then the difference in rotor sizes that I've said. And another important point is the way that they're put together is very different from the human enzyme. So I've been studying with my colleagues the assembly of the human enzyme for this reason in recent times. I don't have time to describe that. We do understand a great deal about how the human ATPase is regulated. It's regulated by a protein called IF1, which will inhibit the hydrolytic action, but not the synthetic action. It's a simple protein, 86 amino acids long. You can see it's alpha helical. 2 proteins held together in a dimer by an antiparallel coil. And it goes into a catalytic site. But actually what we realized subsequent to determining this structure, which was completely alpha helical in crystalline form, we've realized that this part is actually an example of an intrinsically disordered protein. It's not folded at all in reality. And this disordered region interacts with one of the 3 catalytic sites of the enzyme. And then what happens next is the enzyme will hydrolyze an ATP. This closes in, and it folds a little bit more. Another ATP is hydrolyzed, and it folds completely. And so I more or less come to the end of what I want to say. I'll just summarize this, that there are different mechanisms of regulating bacterial ATPases. And I've said that some are reversible, some can synthesize, and we're trying to understand these regulatory mechanisms. But I thought I would just finish again with reference to Max Perutz. So in 1978, when I had this idea of working on how ATP is made, I explained it, what I was intending to do to Max, and he listened very patiently. And then he just looked at me straight in the eye and he said, "John, it sounds like a life sentence." And I thought to myself, that's a ridiculous remark. I'll have this sorted out in five years maximum. Well, 40 years later, this is where I've got to. So this is the actual structure of the entire dimeric bovine complex, which where we are. We're still not at a conclusion. There are many aspects of this process that we don't understand. Now, Max was actually, of course, extremely intelligent, urbane, and he was also humorous in a very dry kind of way. He wrote beautifully. He wrote books about his lifetime experiences, which I can definitely recommend. But he had a number of aphorisms that people who knew him remember. So he said, first of all, discoveries can't be planned. They pop up like Puck. So those of you who know Midsummer Night's Dream by Shakespeare will remember that Puck is a kind of elf and will pop up like Puck in unexpected places. And then another thing he said is, what is known for certain is dull. And I agree with that. And then finally he said, and this I think is the most important aphorism in science, truth always wins. And I think we all hope, for the benefit of mankind, that the same will apply in the wider world. Thank you.